Good afternoon, everyone, and welcome to my seminar on the convergent evolution of phenotypic traits in a weakly electric fish. My name is Jason Gallant, and I'm an assistant professor of integrative biology here at Michigan State University. I'd first off like to thank the organizers for the invitation to speak at this special symposium, and I'm very excited to be participating in my first Great Lakes Bioinformatics Conference. My laboratory is fundamentally interested in the convergent evolution of phenotypes. The ability to predict genotype from phenotype relationships has enormous intellectual, agricultural, and biomedical value. By leveraging instances where phenotypes have convergently evolved, we can gain greater statistical power in understanding how genomes respond to selection, particularly if the underlying proximate mechanisms are shared. Importantly, this helps us infer how repeatable the evolutionary process fundamentally is. This benefit extends not only to proximate causes, but ultimate causes of phenotypic evolution as well. My laboratory studies convergent evolution in electric fish. There have been at least 6 independent origins of electric organs in vertebrates, with most of the species falling into 2 large main lineages, the South American gymnotiforms and the African mormyrids. Electric organs did not only evolve for attack and defense. Thanks to laboratory amplifiers invented in the 1950s, Hans Lissmann was able to demonstrate to biologists that these 2 lineages, the South American gymnotiforms and African mormyrids, were in fact weakly electric. These clades of fish use their electrogenic abilities not just for powerful shocks, but also for communicating with one another, navigating their environments, and finding food. More shocking still about these fish is the remarkable degree of convergence between gymnotiform and mormyrids at nearly every biological scale of organization. These taxa have convergently evolved 2 novel organ systems and exhibit parallelism in the molecular adaptations as well as patterns of gene expression between these organs. At higher levels of biological organization, my colleagues have demonstrated convergent evolution of brain circuits and behaviors, locomotor strategies, and even occupation of ecological niches. At the largest scales of biological organization, these 2 clades exhibit both patterns of explosive diversification. There's a worldwide community of electric fish biologists, and historically, weakly electric fish have served as an important animal model for the neural basis of animal behavior. But there's a growing contingent that appreciates the potential of the system in evolutionary biology as well. The electric fish community, as a result, is incredibly rich in interesting phenotypes, but has historically been poor in genetic resources and essentially non-existent when it comes to functional genomics tools and developmental studies. When I started my laboratory in 2013, we identified this lack of genomics resources and functional tools as a key contribution we could make to help move the field forward. In the ensuing years, I've led the development of nearly all the genomic resources for electric fish listed here, which will serve as a foundation for the work that I'll discuss today, as well as numerous future studies. In addition to this work, my lab has specifically developed 2 key technologies for functional genomics work in electric fish: the application of CRISPR-Cas9 gene editing and the introduction of transgenic material using the Tal2 transposase system. In other words, we now have technologies for doing knock-ins and knockouts in electric fish. For the remainder of my talk, I'll discuss how we're using these new genomics resources and functional genomics tools to understand the evolution of weakly electric fish. Specifically, I'll present 3 short vignettes on different approaches to understanding the substrates of electric fish signal evolution. To introduce this work, I'll start out by considering in more detail one group of weakly electric fish, the mormyrids of Africa. Mormyrids are an ancient group of teleost fish, one of the largest groups of vertebrate species on the planet. When we examine the rates of speciation among different teleosts, mormyrids really stand out from the pack, exhibiting an unusually high level of speciation. Presumably, this high speciation rate is due to the diversity of electric signals that are produced by these fish. This is well illustrated by one genus called Paromormorops, which is a rapidly evolved, geographically restricted genus endemic to West Central Africa. This image represents all of the species of Paromormorops we encountered during a recent 6-week expedition to Gabon in 2019. Striking about this assemblage is the relatively low morphological diversity of these fish, but a striking diversity in the electric organ discharge waveforms, or EODs, shown in yellow. We know that EOD waveforms are important aspects of species recognition during mating, and that these characters diversify at much higher rates than other ecological traits, particularly in the genus Paramormorops. So how do electric discharges vary between one another? One of the things that we can notice right away is that the EODs are incredibly different in terms of their duration. Some EODs are very short, whereas others are very long. If we examine this over the phylogeny of Mormyrids, and specifically the genus Paramormorops, we can see a very interesting pattern. Brief EODs evolved early from long duration ancestors, and several genera of Mormyrids have convergently re-evolved long duration EODs, which is the ancestral state of the family. This pattern is mirrored within the Paramormorops genus, where at least two species have independently evolved long duration EODs from short duration ancestors. What are the underlying mechanisms of EOD duration evolution? Electric organ discharges are produced by a novel structure called the electric organ, which is located in the tail, which is in turn composed of many hundreds of individual cells that we call electrocytes. Electrocytes produce electricity by firing action potentials, just like all other excitable cells. And these action potentials result in turn from the opening and closing of voltage-gated ion channels permeable to sodium and potassium. Importantly, the individual action potentials are shaped by the speed at which these channels open. In Mormyrids, each electrocyte has two excitable faces, the anterior and posterior face, that each produce an action potential. These synchronous discharges of electrocytes sum in time and space to produce an extracellular potential that we refer to as the EOD. The common ancestor of teleost fish underwent a whole genome duplication event, giving teleosts an additional copy of many genes for each one of our own human genes. The vertebrate-specific sodium channel, muscle-specific sodium channel, NAV 1.4, is a great example of this. It's encoded by two distinct gene in teleosts, SEN4AA and SEN4AB. While most teleosts express both copies in muscle, weakly electric fish have independently lost SEN4AA expression in muscle and regained it in electric organ. Along with this shift in expression, each lineage has had selection operate on this protein independently and in parallel to tweak SEN4AA to achieve more rapid inactivation necessary for the production of brief EODs. My laboratory recently demonstrated that this sodium channel is indeed the predominant carrier of sodium current in both lineages of weakly electric fish using CRISPR-Cas9 gene editing. Because of the relatively low pleiotropy of this gene and parallel expression in both lineages of weakly electric fish, it was a useful target gene for demonstrating the feasibility of gene editing in these species. Using CRISPR-Cas9, we were able to induce a variety of insertion-deletion polymorphisms that created null SEN4AA mutations. In this video, we compare the resulting wild-type electric discharge of the F0 crispin to its wild-type sibling, demonstrating the amplitude of electric discharges is considerably reduced. Our collaborators at UT Austin have now discovered the cause of SEN4AA loss in South American electric fish. Together, we were able to identify a highly conserved non-coding element upstream of the SEN4AA transcript in vertebrates. We've removed this CNE using CRISPR-Cas9 gene editing in zebrafish and show that it eliminates SEN4AA expression. We can also create a reporter construct that fuses CNEs from different vertebrates with green fluorescent protein to demonstrate that this CNE specifically drives expression in muscle. Thanks to the development of genomic resources that we've done in electric fish, we were able to compare the sequences of the CNE across vertebrates, including weakly electric fish. We were able to show that this CNE degenerates in South American lineages that have lost SEN4AA expression, but not in Mormyrid electric fish from Africa. This conserved non-coding element contains two transcription factor binding sites, one for TEF1 and another for the transcription factor myogenin. My laboratory specifically applied Tol2 transposase approaches to evaluate whether or not the trans environment in species that have lost CNE3 expression is preserved. To do so, we injected the Danio CNE3 fused with a green fluorescent protein into weakly electric fish embryos. We demonstrate that green fluorescent protein is expressed in skeletal muscle, but not in the electric organ. Together, our results indicate that the loss of this particular conserved non-coding element is responsible for the loss of SCN4AA in at least one lineage of weakly electric fish, but not the other. We've also examined the evolution of potassium channels, now specifically in mormyrid electric fish. In an analogous situation to the sodium channel, the teleos-specific whole genome duplication resulted in two redundant muscle-specific potassium channel genes, KCNA7A and KCNA7B. In mormyrids, KCNA7A expression is lost in muscle, but restricted to the electric organ. This loss is correlated with an ancestral shift in mormyrids from long duration to short duration EODs. During the shift in expression, we see a correlated burst of positive selection. The substitutions resulting from this positive selection specifically target a region of the S3-S4 linker, which we've demonstrated results in the channel opening at lower membrane potentials. This has the effect of actually shortening electrocyte action potentials. Interestingly, further mining of our genomics resources for mormyrids has uncovered a new twist to this very interesting story. As I mentioned before, some mormyrids have independently regained the ancestral long duration EOD. In at least one species, we've demonstrated that this negatively charged patch reverts back to the ancestral positively charged patch. This demonstrably lengthens the action potential and is thought to increase the duration of the EOD. We're currently investigating whether this substitution repeatedly evolves in other lineages of mormyrids that have evolved long duration EODs independently, so stay tuned. Another important dimension of EOD variation is signal complexity, exemplified by this figure here. Mormyrid EODs come in two different types, biphasic, shown in blue with a rising and falling phase, and triphasic EODs, which are shown in red with a small head negative pre-potential followed by the rising and falling phases. Similar to the situation with EOD duration, biphasic EODs are ancestral, but triphasic EODs evolved early. In this case, many genera have convergently evolved biphasic EODs by reverting to the ancestral state. If we zoom in on just the paramormorops, we can see the same situation occurring again. My laboratory has spent much of the last decade trying to understand the proximate and ultimate mechanisms underlying the convergent evolution of biphasic EOD phenotypes. The mechanisms underlying this difference between biphasic and triphasic EODs rest on the underlying microanatomy of the electrocytes. Very briefly, mormyrid electric fish connect their electrocytes to the nervous system through derived structures called stalks. These stalks can either pass through the electrocyte membrane through small holes called penetrations that create this negative phase in triphasic individuals. Alternatively, biphasic individuals have a simpler anatomy that lack these penetrations. Thus, the underlying cause of biphasic triphasic EODs is a major change in the anatomical configuration of electrocytes. Many years ago, I discovered that a geographically widespread species of paramormorops known as Paramormorops kingsleyii is polymorphic for this trait. Entire populations of fish where it occurs in Gabon are either fixed for this triphasic or biphasic EOD and its underlying anatomy. My laboratory saw this as a unique opportunity to uncover the genetic mechanisms underlying the reorganization that occurs broadly across mormyrids. So we embarked on a whole genome resequencing effort to identify causal mutations in these populations. Shown here is a maximum likelihood tree based on hundreds of thousands of unlinked SNPs. This indicates that Paramormorops kingsleyii is itself a microcosm of this phenomenon. Biphasic EODs have evolved as many as 3 times independently from a triphasic ancestor. Notable here is the very close relationship between two populations, Apasa and Bambomo creeks. Based on several lines of evidence, we believe these populations form an active hybrid zone between triphasic and biphasic individuals. This discovery has created the ideal circumstances to perform a genome-wide association study to try to identify causal mutations. Shown here are the results of our GWAS between these two populations, which suggest two very strong associations in very restricted locations in the genome, particularly at chromosome 21 and chromosome 10. These GWAS peaks correspond to a recessive haplotype found in Bambomo Creek in the coding region of the gene spectrin beta 4, located on chromosome 10, and another region of highly associated SNPs upstream of the protein contacted associated protein 5, better known as Casper. Interestingly, the variants associated with spectrin occur in a region just 5' of a myogenic tissue-specific splice variant of spectrin beta 4. When we examine the expression of this gene between the two phenotypes in adults, we see no strong differential expression. This suggests that this region could influence gene expression early in development, when penetrations form, or possibly affect the temporal or spatial distribution of the protein. On the other hand, we demonstrate a strong and highly significant pattern of differential expression of Casper5 between biphasic and triphasic EODs. We can also demonstrate that these proteins are in the correct location, while presently we only have an antibody for spectrin beta 4. In this series of immunohistochemistry images, we see that spectrin beta 4 is strongly expressed in the middle of electrocytes and actually forms a protein ring around penetrations in the triphasic Brettanomyces brachyisteus. Finally, we know that the spectrin beta 4 and Casper5 proteins actually interact. Perhaps best studied is their interaction when they form the nodes of Ranvier, which is another type of interaction between two distinct cell membranes that must be held together. We speculate that these proteins somehow interact to form the penetrations themselves. We're very excited to have identified for the first time a handful of good candidate genes in the origin of biphasic EODs. Both variants appear to be cis-regulatory in nature. So to conclude, I'd like to thank the members of my laboratory, both past and present, who've been terrific colleagues and have contributed to the different aspects of these stories. I'd also like to acknowledge our many collaborators that have helped us both in the field and in the laboratory, particularly Harold Zakon and the rest of his team at UT Austin. Finally, I'd like to acknowledge the generous support that we've received from the National Science Foundation and MSU, who's made this work possible. Thank you very much for your time and attention.